Professor Dr. Mark Zimmer, who works at Conn College and works on bioluminescence, and just recently published a book called The State of Science. And we are going to hear a little bit about the state of science. Go ahead, Mark.
Thank you.
And thank you for coming and talking to us.
Yeah, I wish I could actually be there in person. So it's a small group, so please, if you have a question, I think the easiest is just unmute yourselves and ask the question while you have it, or at the end. But if you feel comfortable, I'd be very happy to answer questions as we go along. So The State of Science is a book that I wrote just before COVID. It got published in COVID, but actually everything that I spoke about is still relevant now, maybe even more relevant. So all the good things about science, like the finding of the vaccine so fast, and all the bad things about science, about fake science and so on, they're there, and we foresaw all of them. So the book is divided into 4 parts: good science, bad science, old science, new science. And what I wanted to do today is maybe talk a little bit about bad science, just because that's more interesting. And there are also lots of things to think about when we think about science being bad. And I would say science in itself is not bad, right? It's how it's being used, misused, or also misrepresented that makes it so bad. So if we look at science and we go back thousands of years ago, we look and we'll see that humans were a tasty snack to the predators that were around at that time. Now, even at night, right, we can walk around and there's nothing going to jump on us and eat us up. We're on top of the food chain now. And most of that is because of technology and science. It's fertilizer, fire, building houses, cars, and things like that. And we're sort of become the dictators of nature in some ways. We're completely in charge of what happens in nature. And we'll see just at the end of this talk, we're actually a lot more in charge than we've ever been before, because now we're actually in charge of evolution. In Devlin, genetics is no longer a thing. We can control genetics completely. So if you look at our sort of record, if there were no humans on Earth, there'd be about 1,000 times less extinction than there is now. We're 1/100th% of the mass of all living things on Earth. So 100th%, and yet we've wiped out 83% of all the mammals. So giving us this scientific power is something that comes with responsibility, and maybe we aren't dealing with that responsibility very well. And what we see is that science is growing really, really fast, faster than any other field. And in some ways, this is good. If you look at the economy, our economy is probably science-driven now. A couple hundred years ago, economy was driven by agriculture, and the top leading countries, England, Germany, Holland, well, Germany less so, Spain and Portugal, all had a lot of agriculture, and to get more agriculture, they colonized lots of places. After a while, though, agriculture was no longer the driving force for economy, industry took its place, and now science is really driving the economy. If you look at the biggest companies, there are things like IBM, like Tesla, Baidu, Pfizer. They're all big science companies. So that's good about the growth of science. But the other thing that's not so good is with the growth of science also comes the growth of power. We saw this with nuclear power. right? Nuclear power was very, very potent. We could do lots of things with it, but there's also some danger associated with it. And in the book, in the news section about science, I talk about 4 new techniques that are really going to change the world. One of them is gene editing, CRISPR. The other one is something called optogenetics, which I work on as well, is where you take light and you can make neurons fire. So basically, you can control the brain by making individual neurons fire with just a beam of light, a laser beam. You can see that's got a lot of power and a lot of ethical problems coming up the road with that. And then the other thing is I'm a computational chemist, so I use computers to calculate all these things. And it turns out that the big new development in computers is deep learning, where the computer takes a lot of data and uses that data to teach itself how to do things. And again, the more data one has, the more computational power one has, the more one can do with it. So these big companies like Facebook and Google are getting all of this power. And Google can already predict where there are outbreaks of diseases before the World Health Organization knows it. So for example, dengue fever, it's spread by mosquitoes, and people are looking for the symptoms of certain malaise, and Google can immediately see, oh yeah, there's an outbreak of dengue and it's spread this far around a certain city. And so with having this information comes a lot of power. So what I thought I'd do is I tell you a little bit about what I do in my research. And in the book, I've called this old science. And then I'll show you some of the new science. And with the new science, talk about some of the dangers. So let me see if I can share my screen. Where have you gone to? There. I hope you can all see that. I don't need that. Don't need that. One thing that I did want to say about science is people have this misconception of science. A lot of people think that science is like math, where you've got a proof and that proof is 100% known and that's the way it is. But in science, I think a good analogy for science is a puzzle, especially now in the COVID times where I think a lot of us have probably been making puzzles. You can think of nature as a huge, big set of puzzles, beautiful, big, three-dimensional puzzles. We don't have all the pieces, and nor do we have a picture. So it's really, really difficult. So when you get something new, like, for example, COVID, first thing we learned is it belongs to the coronavirus family. And then we know something else about the other sicknesses like SARS and MERS. And so we can learn slowly about COVID. And I like to think of all these new things as new pieces. People work in the lab, people work in hospitals, they find something, they publish it. Once it's published, that's a puzzle piece. And if you have a very important puzzle piece, it gets published in a very good journal, but also that means other people can take their puzzle pieces and put it with yours, and a picture starts emerging. So here we see a puzzle of a bear, and I think most of us can see that it's a bear. And as the pieces come together, we can see more and more of this bear. We can see a lot of its right-hand side, but we can't see much of the left-hand side. And that's how science works. Somebody does some experiments, comes up with a piece and says, wow, you know, this piece looks like a sail of a yacht. And in the back, there's some mountains. Maybe this is a yacht in a harbor in the Mediterranean. and you publish exactly how you got this piece, what the piece looks like, but then typically you also hypothesize a little bit. And you say, okay, well, this is what I think it is. Somebody else does some experiment and theirs fits in beautifully next to yours and overlaps a bit, so they publish it, and now they say how it fits with yours. But after a while, you find out that you've been making a hot air balloon, and it wasn't a sail. And so the science gets corrected. And as more and more pieces come together, we can see that, oh, for example, this is turning out to be a bear. And so science is sometimes wrong, and that's okay, because that's sort of the process of doing science. As we do more and more of it, we find out more and more of the science. It's a problem when people put in false pieces. You can imagine if you've got a puzzle and somebody puts a piece in that doesn't belong, then it gets really difficult to put it together. So false science is a real big problem here. When you read about science, you typically read about it in retrospect. It's like history. And what you see is that straight path that goes from the beginning to the end. And so you can see how the end result came about, but you don't see all the paths that people tried that didn't work. And so, for example, with Covert, there are a number of theories that went out there by legitimate scientists that have since been proven to be wrong. But I think we need to remember that that's just the way science is. So let me tell you a little bit about what I do, and then we'll see how it's relevant in just a bit. So I'm interested in bioluminescence, animals that give off light. I'm particularly interested in this jellyfish. It's called Aquara victoria, or the crystal jellyfish. It's about as big as my hand. In the bottom here, in the umbrella, it's got about 300 of these photo organs that give off green light. There was a Japanese researcher who caught about 1 million of these jellyfish, isolated the materials in these photo organs to try and figure out what the chemistry was. It took him about 30 years to do this, mainly because this worked in a completely different way. What happened was when you agitate this jellyfish, just sometimes it lights up. And when it lights up, so you've got to agitate it a couple of times. Doesn't want to be agitated. There it gets agitated again. And then what it does when it lights up, come on. It gives off calcium. And the calcium binds to a protein that is only found in this jellyfish. And this protein is called aquarin. Now, aquarin, when it binds to calcium, gives off beautiful blue light. But if you look at the jellyfish, you never see blue light. And the reason you don't see the blue light is that blue light energy is given off to another protein, and this is why it was so difficult to figure it out, called green fluorescent protein. What this does is it takes this blue light energy and changes it to green light, which has less energy. So it absorbs all this blue light energy, changes to green. Now I'm going to tell you a little bit about this green fluorescent protein, which is what I work on. But before that, I sort of want to tell you something that really bothers me. So I work on aquarin. I know Doug Prasher, a friend of mine. He was the one who started working on this in his postdoc in the 80s, quite a while ago. And he found the gene, the DNA, to make aquarin in the jellyfish. And was also responsible for then being able to put this into bacteria, so the bacteria make aquarin. And it turns out this is really useful because then you can make lots of aquarin and you can inject it into a muscle, for example. And when the muscle contracts, and there's a lot of calcium, the calcium will bind to the aquarin, and the aquarin gives you blue light. So you can have a sensor for calcium in muscles, and this is typically done in model organisms, and even in the brain. So you can put aquarin into the brain, and if the neuron fires, the concentration of calcium goes up and it gives off blue light. So you can actually see which neurons are lighting up. So that's the research that Doug Prasher did with aquarin. Now, one of the things that he found out was that aquarin is non-toxic. So he did a lot of experiments and found out, amongst other things, that it's non-toxic. It's also inexpensive because you can make it from bacteria. But it's a protein. It's named after the jellyfish, which is called aquaria. And so if you eat it, like most of your proteins, right, it goes down your stomach, it gets broken down by the stomach, it gets digested, breaks down into the amino acids. The useful ones are kept, but none of it ever goes to your brain, and none of it certainly crosses the blood-brain barrier. So the reason I'm telling you this, and the reason this drives me crazy, and this is talking about bad science.
I'm Norm.
I'm Saz.
And we live in Columbia, Missouri. We do consulting, but we also write.
We take care of ourselves constantly. It's important. We walk 3 to 5 times a week, a couple miles at a time.
We've both been taking Prevagen for a little more than 11 years now. After about 30 days of taking it, we noticed clarity that we didn't notice before.
It's still helping me. I still notice a difference.
Prevagen, healthier brain, better life.
So how many of you have seen this ad or a version of this ad? So it typically plays on the news, and pretty much every other day on CNN, on Fox, CBC, NBC, all the news and all the football games and sports things. Prevagen is aquarin. It's that protein from the jellyfish. And the ad, a little thing like this costs $50. The ad makes it sound as if when you take Prevagen, when you eat aquarin, you're going to remember things better than you did before, right? But at the bottom here, there's that little statement. These statements have not been evaluated by the Food and Drug Administration. This product is not intended to treat, cure, or prevent any disease. So one of the problems with science is that science is getting so big and growing so much that it's very easy for companies like this to prey on unsuspecting people and sell things like this. And there are many, many examples of this. It's also very easy for people to say, you know what, I don't understand the science, so I'm just going to believe what I'd like to believe. I'm going to believe what my social group believes. I'm going to believe what's part of my social identity. That could mean that I believe that genetically modified organisms cause allergies and are unhealthy. It could be that I believe climate change is not real. Both of those are two political extremes, but both of them are unscientific beliefs. So, so a lot of people, I find it particularly irritating being at a university and seeing all my colleagues who will do this, who will say, oh yeah, but the science is so difficult. I can't read it up, and so don't worry about it. I'll carry on taking charcoal milkshakes. And even though as a chemist, I can tell them exactly how dangerous it is and why. So that's sort of one of my bugbears. But let's go back to this jellyfish, right? So we have the jellyfish. It gives off calcium, aquarin, gives off blue light. The blue light gets absorbed by green fluorescent protein, and the green fluorescent protein then gives off green light. Now, proteins are tiny, right? We can't see proteins. They're so small. But it turns out that instruments and our eyes are very good at detecting light. Now think of the stars. During the day, we can't see the stars, but at night we can see all of them. Think of a firefly. You can see a firefly flying a football field away, and you're just seeing a small fraction of its bottom lighting up, but we can see it. So the idea was, could we take this green fluorescent protein and use it like a torch, a flashlight? Could we stick it at the end of any other protein, and then if we shine a blue light on it, does it glow green? So that's sort of here. If you think about DNA as a huge recipe book, and each recipe is called a gene. So this is the gene to make this protein. It's just the recipe how to make this. You can put at the end here, before there's what's called the stop codon. This is just a message saying, hey, this is the end of the recipe. If you could sneak in the gene for green fluorescent protein from the jellyfish, sneak it in here, then when this protein's made, it doesn't stop making it. Instead, it carries on making green fluorescent protein. And now, if you shine a blue light on here, it glows green. So now we've got a way of tagging when proteins are made and where they go to. So this is, I calculate what happens when you change part of this green fluorescent protein and how to make different colors. So that's what my research is all about. And it's funded by the National Institute of Health because it's very, very useful. Let me show you an example, a very, very relevant example. COVID, right? We know it's caused by a virus. We now know that it's caused by a virus that enters the respiratory system. And it turns out that if we're looking at respiratory diseases, ferrets are really, really good models. For the flu, for SARS, for MERS, for COVID, ferrets are the best model. Now, if you want to see what does the COVID virus do, well, what have they done with it is they've genetically modified the virus. So one of the proteins in the virus, the gene for that protein, has green fluorescent protein in it. And then they've sprayed the virus into the air around the ferret. The ferret breathes it in, gets infected. And this here is a cross-section now of the bronchial tubes. And these are the ciliated cells. So the cells that have little hairs on the top. And you can see they've all been infected by the virus. So the virus has entered these cells. When it enters these cells, it multiplies. And when it multiplies, new proteins are made. All of these new proteins have green fluorescent protein. Shine a blue light on it, and you can see exactly where it is. And then you can also see with progression of this disease, what happens to it. Does it go down? Does it go up? And how does it proceed? So this is just a really neat way of tracking things. This is quite a famous photo in Europe, but somehow it didn't make it in American media. These two pigs are clones. So actually the genetic material to make these two pigs comes from their mother's ear. So these two pigs and the mother are genetically identical. We can think of them as triplets. But this one has been genetically modified so that it makes yellow fluorescent protein in the snout and in the trotters. So this experiment cost a couple of $1 million, took about 5 to 6 years to do, and 5 PhD students. It was a humongous piece of work. So it was done about 10 years ago. So why would somebody want to do this? Well, the idea is that pigs grow in all kinds of sizes, but you can get pigs that are just the right size that their organs will fit into humans. Mechanically, pig organs work very well in humans. But there are a couple of problems, right? Our immune system rejects them. Well, what if we had a pig that the day the baby is born, we genetically modified this pig, so this pig and all its offspring have the proteins and sugars that our immune system looks for when it rejects something. So this pig is genetically modified. It's still a pig, but it's got about 100 human proteins and about 50 human sugars. And not only human ones, but exactly your sugars and proteins. So then if you take a heart from this pig, your immune system looks for all these markers. They're all present. In fact, they all look perfect because they actually came from you, and it doesn't reject them. So that's the whole idea of this experiment. Okay, so let's jump a little bit forward. So this is what I'd call old science. Let's go to new science.
The Royal Swedish.
Let's just go around quiet for a while. So I'm a science geek. I'm very, very interested in the Nobel Prizes. 2008, my wife and I were invited to go to the ceremony, to attend the ceremony. So that was really, really interesting. And so every October, I wake up early and I listen to see who has won all the Nobel Prizes. This was this October, and this was the announcement of the Chemistry Prize.
The Royal Swedish Academy of Sciences has today decided to award the 2020 Nobel Prize in Chemistry jointly to Emmanuel Charpentier and Jennifer Doudna for the development of a method for genome editing.
So I was ecstatic for a number of reasons. One of them is this gene editing was one of the 4 techniques that I'd predicted were going to be big in my book. So it was nice that that was already there. But another one was that the prize went to Jennifer Doudna and Emmanuel Charpentier, 2 women who richly deserve this prize. It turns out if you go back in history and you look at the 621 Nobel Prizes that have been awarded in chemistry, physics, and medicine, 3% of those went to women. So having 2 women do this and do this for such a cool technique, I thought was really, really rewarding. So I also liked this because after the announcement, the chair of the chemistry Nobel Committee came on and had this to say.
The enormous power of this technology, it means that we need to use it with great care. But it's equally clear that this is a technology, a method that will provide humankind with great opportunities.
So what he is saying is that this Nobel Prize comes with a warning label, right? Beware, this is a very, very, very potent technique. So I thought I'd just talk a little bit about what CRISPR is. And if you go back to this, my piggies, right? These pigs were genetically modified. And I told you that it cost a fortune and took a very, very long time. Well, with CRISPR, the gene editing technique that Jennifer Doudna and Emmanuel Charpentier derived, you can genetically modify little pigs like this one for tremendously less money and effort. This pig was genetically modified. It took a couple of months and it took a couple of $100,000, but they genetically modified 69 proteins in one shot. So the previous one cost millions of dollars, took many years, took many PhD students. Here we've got just very quickly, and we can do lots and lots of genes. The easiest way to think about this is to think of before CRISPR, when we genetically modified things, it was like using a typewriter. If you had something, you wanted to change something, you had to go and find the page by going through everything and then change with the typewriter one thing, and you probably had to retype that whole page. CRISPR is like a word processor. We can have CRISPR go through all the DNA, make all the changes we want in every gene. We can delete things, we can add things, we can do things much, much quicker. So quick and easily. So if I had a student who wanted to genetically modify a ferret, for example, 10 years ago, that would have taken years and years and years and would have cost tens of thousands of dollars. Now it'll cost less than $100. I just type out the sequence on the computer, I send it to a company, they send me the CRISPR with the lead that finds the place that I said I want to change, and it automatically finds it and changes it. So it's become a lot easier to genetically modify things. This is from an article that's coming out in this weekend's The New Yorker. It's written by one of my favorite authors, Elizabeth Colbert. And in it, she sort of describes some of the things that have been done with CRISPR. So with CRISPR, biologists have already created, amongst many other living things, ants that can't smell, beagles that have put on superhero-like brawn, pigs that resist swine fever, macaques that suffer from sleep disorders, coffee that contain no coffee, beans that have no caffeine, salmon that don't lay eggs, and mice that don't get fat. So those are some of the things that you can do with CRISPR. Here's another really good thing you can do with CRISPR. There are about 10,000 diseases that are single gene diseases. So they're caused by a defect in a single gene. The problem really, of course, is how do you change them all in all the cells in a human person? But there are some diseases that are easier to treat than others. And Victoria Gray had sickle cell disease. So in sickle cell, what happens is the red blood cells, which are sort of donut shaped, can collapse. And when they collapse, they become sickle shaped and they don't transport oxygen throughout the body. This leads to sore arms, sore legs. If the oxygen doesn't come to the brain, headaches, can lead to comas. It leads to early death. It's a disease found most commonly African Americans who come from West Africa, because it actually turned out to protect their ancestors from malaria. Victoria Gray had sickle cell disease. She had to go to the hospital about 12 times every year, suffering from chronic pain and fainting and so on. Now she hasn't been for the last 2 years. What they've done with her is they take out blood plasma from her out of the body, and then they genetically modify all the hemoglobin genes. Hemoglobin is the part in the red blood cells that are red, and they replace that gene with a gene for fetal hemoglobin. So hemoglobin that's normally found in babies, that's found in the embryo, and in babies it's there for about the first 3 or 4 months, and then they start using adult hemoglobin. But fetal hemoglobin is much, it's a strong oxygen binder, and it doesn't have the sickle cell mistake in it. And so then they inject that plasma with the modified hemoglobin gene. And so Victoria Gray, as an adult, makes fetal hemoglobin, but is completely healthy. So this trial on one person at first has now been extended to more people. So in some diseases in which one gene is wrong, and especially if that's something we can take out the body, genetically modify, check, it's easy to do. Another place where these genetic modifications are in eye diseases. Apparently it's a lot easier to localize that and to get a virus to make all the changes you want. Okay, so this is a really potent and useful technique what is the problem? Well, there are a number of problems. One of them is that just like there are about 10,000 diseases that can be modified, there are also 10,000 genes that occur in very rare populations that, if they were placed into an in vitro fertilized embryo, would make that embryo a superior type of embryo. So there is, for example, a gene that's very common in marathon runners. It's responsible for extra endurance. So you could take an in vitro embryo, genetically modified, so that baby has this gene and will grow up with extra endurance. There are all kinds of ones. There's some that are responsible for stronger bones. So what we can see here is a place where we start getting designer babies and can actually design babies. So that's obviously a big problem. Another problem is one that I'd alluded to before, which was that we're now in charge of evolution, and that Mendelian genetics is no longer a thing. So if we look at normal inheritance here, we have two fruit flies. One of them is this blue one and the other one is red. When these two fruit flies mate, there's approximately a chance that half the offspring, this one, is red, and the other half is blue. The blue one will mate with the blue one, so all the offspring here will be blue. This red one will mate with a blue one, so half of the offspring, this one is red, and the other half is blue. And so you can see nothing really changes much. But then you get something called a gene drive. With a gene drive, what you have is you have CRISPR, that, remember, the CRISPR is the word editing program. You put in into this red fly the CRISPR that says, okay, whenever you find a gene for anything that isn't red, replace it with a red one. So it's like putting into the baby a little photocopier that replaces the gene that's already there with the red one. So this red one mates with the blue one, both offspring are red. These both have the gene drive, so all their offspring will be red. The first of these experiments were done about 4 years ago, and calculations have been done if those fruit flies that were genetically modified with a gene drive in San Diego had escaped, all of continental US would now have yellow fruit flies. They would have changed color by now. So what can we do with this? Why are people so interested in it? Well, it turns out that probably the first experiments, unfortunately, will probably be done in Burkina Faso, which is in Central Africa, has a huge malaria problem. And Imperial College has got this program where they've got mosquitoes with a self-destructing gene drive. So what these mosquitoes do is the mosquitoes, female mosquito lays eggs, and she's been genetically modified so that when the eggs hatch, all the females grow to be larvae, but no further. They never mature to a fully grown mosquito. All the males grow to be fully grown mosquitoes with this gene drive inheritance, that when they have reproduced with a female, that female will lay eggs. All the eggs, the males will have the gene drive, the females will never grow older than larval stage, and so the females just disappear, and then the whole population collapses. So at the moment they have these massive mosquito camps where they're doing these experiments in the field, but they're still trapped in big warehouse-like structures. But they want to release them. Now, what does that mean? Well, that means once you've done this, there's really very little to stop it, because everything will have this gene and they'll be everywhere. So it's quite easy to stop this when you've got them trapped in a warehouse. But if they're out in nature, how are you going to catch all the mosquitoes? Because even if just one is left, that one will carry on passing its gene through. So this is if you think of this as an environmental chemist, this is a beautiful pesticide. It's species specific, right? Because the Anopheles mosquito is not going to breed with any other type of mosquito, just with other Anopheles. And so they're the only ones that will die. This is not going to affect butterflies. It's not going to affect bees. It's not even going to affect other mosquitoes. But for the first time, really, humans can wipe out one species. One person can with one little experiment. And of course, you can think of this, it turns out the biggest funder in this field is DARPA, which is a military-based American branch. They're probably thinking defensively, but still the fact that people are thinking that something like this could actually be used in biological warfare is very, very scary. So this is sort of the big strength of all of these. The power comes with danger. It's one of the big things I wanted to say. Marshall Nernberg, he won the Nobel Prize in 1968. He had this very famous quote that when man becomes capable of instructing his own cells, he must refrain from doing so until he has sufficient wisdom to use this knowledge for the benefit of mankind. And I like this quote for two reasons. One is we've reached this point. I don't think we have sufficient wisdom. Also because this is a very much a 1968 quote, right? When man becomes his own cell, he must refrain. So that the people who found this were two women who got the Nobel Prize for this, I think is also just sort of sticks it in a little bit. I like that. So, but yeah, I think this is one of the things that really we don't talk about enough, right? So I thought I'd go through this quickly. I've got lots of other things I can talk about. So this is just a cover of the State of Science. If anybody is on Twitter, I've got a Twitter account called @LightUpScience, where I talk a lot about science and bioluminescent things. And I'd love to answer questions, many questions. Let me.
I had a question.
Yes.
My question, Dr. Zimmer, was that.
Mark, please.
Okay, Mark, that you said something in the very beginning about how the genes, I guess, in the pigs in the very, very expensive study were made just for you, like for humans in general, or do they try to make them custom for each person? Which did you mean?
No, the specific individual.
Wow.
So the idea is a child is born, and there will be a set of pigs at some pig farm that has been genetically modified for this specific child. If this specific child at some point needs a kidney, a lung, of course, right? What we're getting here.
How does that work time-wise though? Because you don't have time. A child is sick and has needs a kidney, and how many years does it take to generate that gene for the pig or to help the child? How does that work?
So the idea is that this pig and this multiple generations of the pig will always be a backup. So most of the transplants don't happen in younger years. Most of the transplants happen in older years, right? My age is when people start having to think about new liver, new kidney, and new heart. And then there will be a pig that's just there for us. One of the big problems with this is really all of these sort of things that we're thinking about that are going to change medicine are going to be very expensive. And so be more available to rich people than poor people.
Mm-hmm.
I also had a question. This is Kathleen. On the mosquitoes, my understanding is that, birds and bats eat their body weight every day in mosquitoes. So if you wiped out all these mosquitoes, have they looked at which species would be affected that rely on those mosquitoes for a food source?
So the two sort of things that they say there is one is that the Anopheles mosquito, that's the malaria one, and also the Aedes one, that's the dengue mosquito, are both invasive species in most parts of the world. So what you'll be getting rid of is just an invader. The other thing also is there are thousands of mosquito species, and another species will replace those species. And so hopefully that would be a species that doesn't carry a disease. But that's the argument that you would get from the people who are doing that research. And I think...
Sorry, that was me. So if, for example, the Chinese carp is a huge problem in Michigan, can that be eliminated in the same way?
Oh yeah, so
is anyone thinking about it?
Lyme disease. Kevin Esfelt, he's at MIT. He's really interested in stopping Lyme disease, and he's starting to work on Nantucket and Martha's Vineyard because both of those are island systems, so nothing can really escape very easily. But yeah, he's doing it with a lot of public support. So he has a lot of meetings with people, and I can't remember what it's called. There's this big ugly toad that you find in Australia that's toxic. But the animals aren't, because it's an import, the animals in Australia, the koalas and the kangaroos, think it's a snack because they haven't seen it before. And when it gets eaten, it releases this toxic slime. And that's another species that they're looking at right now. I haven't heard about the carp, but I would think that that's certainly doable.
You had,
Yeah, you're on.
am I on?
Yeah.
Yep.
Yeah, you had mentioned that there were 4 techniques that you thought were going to be ascendant. And then I have another question. There are 4 techniques that were going to be ascendant. And you mentioned CRISPR and the one that sort of lights up brain cells. I don't remember. Did you mention the other 2?
So deep learning.
I mean, it's like AI, one of them?
Well, so this branch of AI called deep learning.
Deep learning, okay.
Which is, so just for the calculations I do to calculate the shape of the proteins, these calculations are based on physical equations. I would say there have probably been 10,000 people over 50 years who've come up with these iterations of this program to try and calculate the shape of a computer. And every 2 years we have a competition where all the programs try and see who can calculate the shape the best. This year it was won by a deep learning Google entry that didn't use any physical equations. That was just humongously better than we've ever expected. It was like 20 years in advance we would have thought, well, maybe that's what we'll get. And this is the same type of artificial intelligence part where the computer beats the biggest chess players and can win at Go and poker. So that's another one. And then the 4th one, actually, I don't think it'll change our lives. I think it's just a beautiful science, is gravitational waves. So in Washington State and Louisiana, we have these 2 lasers that go out 90 degrees from each other that are 4 kilometers long. Both of them are 4 kilometers long. And these waves come from collisions of black holes billions of years ago. They hit the Earth, and the Earth stretches and compresses by, a millionth and a billionth of the length of our hair. But what that does is it takes these 2 lasers and throws them out of sync. And then we know that these gravitational waves travel at the speed of light. So the distance between the one in Louisiana and Washington State is a couple of seconds in speed of light. So if we have a deformation in Washington and then 3.1 seconds later, the same deformation in Louisiana, we know it's a gravity.
If he froze.
So that, I just thought that was a very cool technique.
I have a question about applications of your research, fluorescence, in oncology, because if I understand, it is used very widely now. Could you talk a little bit about it?
Well, the very, very simplest one. So most of this, you can think of it like a microscope. It's a tool, a technique to see what's happening. So in oncology, we have cells that are growing uncontrolled and they metastasize, they move through the body. If you could see how they're going through the lymphatic system or the blood system and seeing where they're growing somewhere else, that would be very useful. So you can have I actually had a couple of, as a pet, green fluorescent mice. So every cell in the mouse is green fluorescent. And then you can implant a human breast cancer that has been genetically modified, so it's red fluorescent, topically on the breast of the mouse. You can now, in the living mouse, see the fluorescence. And then when a single cell breaks off and goes into the bloodstream of the lymphatic system, you can actually see and follow it in the living mouse. And then you can see, oh, here it gets stuck, but it doesn't carry on growing. Here it gets stuck and it starts growing and you get metastasis. Then you can take different drugs and you can see what happens. So you can see the tumor growing, the blood vessels growing into the tumor. And because they're different colors, right? You can see everything that's green belongs to the mouse. Everything that's red comes from the tumor. And then you can give them some drugs and see what is it stopping. Is it stopping the growth of the blood vessels? Is it stopping metastasis? How does it work? You can all do this in one mouse. Before fluorescent proteins, you'd have to do this in hundreds and hundreds of mice, cut them, dissect them, look for the growth and see how much there is. In the operating room and so on, probably one of the first places it'll be used is also with oncology. It turns out there's certain bacteria that collect on the margins of cancerous growths. And that's because there's a different oxygen level there. And so if you take these bacteria and genetically modify them so that they're fluorescent, what you can find after surgery is parts of a tumor that maybe haven't been excised properly because they'll glow. And so they've done this with rats and mice and bunnies now. And it works apparently incredibly well. They can find parts of cancerous growths that they didn't find before. And so the surgery is just so much better. And so just a day before surgery, one would be injected with these bacteria would find their way to the tumors. And then wherever the growth has been opened up, afterwards you shine on the blue light, you can see it. So, I mean, I'm a chemist, right? And this is my interpretation of all of that.
Is there anybody that oversees the ethic aspect of these experiments?
So in England, it's very well organized. In England, if you want to genetically modify something like a bacteria, you just fill out a form online. If it's a bigger organism like a bunny or something like that, then you've got to go to a committee. In America, it's organized if you're getting funded by the government. So if it's NIH or NSF funding, then there are rules you have to go through an animal use committee. If you're doing it by yourself, you can do whatever you want. America is one of the most lax countries. We like to think that we aren't, but we are. And most of it is because, well, the government can't get its act together, that both parties can't agree on what they want to do and how much they should interfere. So most of this relies on individuals. I think we probably most heard about the twins in China that were genetically modified with CRISPR. It actually turns out that the scientist who did that got fired from the university in China and was thrown into jail for 3 years. But the person he got his PhD from at the University of Kentucky, who was probably the mastermind behind all of this, is still at the University of Kentucky. And so, yeah, the pig was a clone. In theory, we could make a human clone, and it's not illegal.
We got frozen out and reconnected. I just want to mention that the invasive species phenomenon is absolutely fascinating. Glades, the invasive species is the Burmese python, of which there are thought to be over 100,000 in the Everglades, and they've wiped out 95% of the mammals in the Everglades. And there are a couple really fascinating, because if it's an invasive species, it's hard to say that the Burmese python contributes to the Everglades ecosystem. It's ravaging it.
Yeah, there's an author called Carl Hyacinth. He writes these over-the-top, funny crime stories. And in one of them, there's a Burmese python that escaped in one of Donald Trump's hotels and causes havoc there. It's very, very funny. But yes, the Burmese python, of course, wouldn't be quite as easy to control as the mosquitoes, because I presume their breeding takes much longer, right, to get from one generation to the next to the next. So that's not going to be quite as simple.
I would just like to say that this CRISPR technology scares me. When it's used therapeutically, it's great. But if it's used unethically, like perhaps creating a militaristic society, can you use it? Can it be done?
Well, I don't know about a militaristic society. That's where optogenetics, the other technique that I was talking about, that could certainly, with optogenetics, they've done experiments where they've actually converted mice and made mice more aggressive. But with CRISPR, I think that the big danger is just for the rich, not just getting richer, but actually getting better. That the rich will now be able to be genetically improved. And Yuval Harari sort of talks about Homo superior, that we can maybe get a new species of rich people who've been genetically modified with improved bodies, improved endurance.
Then we have issues of social justice.
Yeah, and eugenics nearly, right? It's just coming back to something like that. There are so many moral issues and difficulties and dangers with this. And I think it's going to be very difficult to control, because these fertility clinics are going to pop up all over the world. And so if one country closes it down, they're just going to move to another country. And it's going to be like medical tourism. People are going to have in vitro fertilization in country Y because they know there that they can have it genetically modified.
There's also, I think, a danger in deep learning to perpetuate stereotypes or prejudices that exist, because the deep learning is done with the data that we have. So there was an interesting example in Google Translate where Turkish does not have gender. So English translated into Turkish gets translated so the Turks understand it just fine. But since this was done with deep learning and the examples for example, physicians and politicians and whatever tended to have the, and athletes tended to have the male gender, and housekeepers and cooks and nurses tended to have the gender she. So when it goes to Turkish, it's fine because Turkish doesn't have gender. But when the Turkish gets retranslated into English, all doctors are he, all nurses are she. Because the examples were overwhelmingly in that thing. So prejudice gets sort of, I don't know, blown up.
Yes, so my daughter-in-law works for a bank, and she's an applied mathematician, and her job is to find, so the bank actually decides on who to give loans using deep learning. And the deep learning is based on old loans, but old loans were given mainly to white males. And so African-American, Latinx women didn't get that many, so the information isn't that good. So her whole job, this is all she does, all the time she works, is to try and find this sort of bias that the programs have and change it. Yeah. So yeah, that's one of the big problems. The other big problem is just who has this data, right? The amount of data that Google has on us, data that we don't even realize they have. I have one of these silly watches. I mean, it collects all kinds of information from me, and goes to a computer. Maybe one of the interesting ones was, there was a study, again, using deep learning. I can't remember what the number was. I think it was 12 million phones. And what they did is they tracked all these phones, and with COVID, they looked at the movement. And what they found was that phones in areas that were socioeconomically disadvantaged moved more. And when they looked at where they moved to, they moved to places where there were jobs. When you went to look at the upper class neighborhoods, the phones didn't move. People could stay where they were and weren't exposed that much to the virus. But just to think that there's somebody who can get the cell phone information from 12 million people and figure out when they went to church, because it was, they found all the churches, they found all the shops, they've found all of this data. So in Science, there's this paper with all these maps moving around. But of course, nothing has your name on it, but it's still a little scary to think that there's all this information.
They actually did that in Israel. They used phones to contact trace.
Oh.
They got a permission to go into people's phones and contact trace for the virus, which,
there is an app in the United States that does the same thing with permission.
Yeah, so that it wasn't, and once you open this door, it's very, very hard to close it.
Yeah, yeah.
Well, this has been an absolutely fascinating talk and a fascinating discussion, and I want to thank you for agreeing to do this. It was really wonderful.
Oh, enjoyed it.
Yeah,
Nice
it was really wonderful.
And tell your friends if anybody wants to come. We are welcoming everyone.
Absolutely. Thank you so much.
Thanks.
Thank you, Mark. It was really, really great and interesting.
Good night, everybody. Thanks for coming.
Thank you.
Yeah.
Thank you.
Wonderful.